to live forever. Imagine the classic vampire, mysterious, immortal, and sustained by the blood of others. It's a chilling idea rooted in folklore, but what if there was a grain of scientific truth in it? What if taking blood from someone younger could actually help extend your life? It sounds like something out of a horror story, but science has started to explore something surprisingly similar through a process called parabiosis. In studies in mice, researchers connected the circulatory systems of an old mouse and a young one. What they found was astonishing. The old mouse began to show signs of rejuvenation. Its muscles grew stronger, its brain function improved, and even its organs started behaving more youthfully. The younger mouse, in contrast, began aging more rapidly. These effects suggest that something in young blood might have restorative powers. Of course, parabiosis in humans would be highly unethical because we can't sew people together. So another alternative that scientists are looking into is plasma infusion. Plasma is the liquid component of blood, consisting of proteins and hormones, as well as other signaling molecules. The idea is that these factors could help reset or refresh aging cells, offering similar benefits without invasive procedures. This line of research ties into two major theories of aging, the first being oxidative damage. According to the oxidative damage theory, our cells constantly produce reactive oxygen species, unstable molecules that can damage DNA, lipids, and especially mitochondria, which are crucial for energy production. Over time, this damage accumulates, leading to cellular dysfunction and the gradual decline that we associate with aging. With young blood or plasma, it is believed that the cells that have been damaged are replenished with the young blood, and this can prevent the act of aging. The second theory of aging, replicative senescence, focuses on the limits of cell division. Every time a cell divides, the telomere cap gets shorter and shorter and shorter, and then when they become too short, the cell can no longer divide and enters a state of arrest known as senescence. These senescent cells build up on tissues and contribute to aging and inflammation. Together, these theories explain why aging happens and why something like plasma therapy might help counteract it. So, let's dive into some research on this topic.
So this study experimented parabiosis in mice, which is the surgical union of two animals, usually one old and another young, to allow the sharing of blood circulation. In this case, a wild type and a GFP expressing mouse were joined, and the goal of the study was to improve parabiosis by joining joints and body cavities. As with other parabiosis studies, scientists desired in their result that the sharing of blood would boost the mouse's antigen defenses and so therefore improve its immune system. Using flow cytometry, the scientists were able to verify successful equilibration of the shared blood system. Blood from the wild-type mouse revealed chimerism as indicated by approximately half GFP positive and half wild-type blood cells. A limitation of this procedure was that it was pretty invasive, and so therefore the scientists had to take great care to prevent infection. So this research article has a group of researchers investigate a whole organism level of aging regulation. At the extracellular level. And the way the researchers wanted to investigate extracellular mechanisms of aging was via parabiosis. Investigating parabiosis with mice, they found that parabiosis slowed down aging in older mice. Parabiosis conveyed evidence attesting to the existence of signal molecules in blood plasma that regulate aging. However, aging actually sped up when applied to younger mice. And this was a large contradictory result and thus a concession within this paper. And another concession that the researchers admit is that research in parabiosis is far from complete. Specifically, they claim that more investigation must be done to reveal the epigenetic factors that might influence blood plasma's effects on aging, and so therefore might weaken or bolster parabiosis's effects.
So, the study by Tripathi and colleagues set out to test a simple but powerful idea. Could young blood, or more specifically, young plasma, actually reverse aging-related damage in older organisms? Building on early parabiosis research, where the circulatory systems of young and old mice were connected, this study tried a less invasive method. Instead of joining bloodstreams, they injected plasma from young rats into old rats 3 times a week for 4 weeks. They wanted to see if this plasma could reverse the biomarkers of aging, especially those related to oxidative stress, a major contributor to cellular damage over time. Now, as shown in this figure, figure 1 measures oxidative stress and antioxidant capacity. In panel A, researchers measured RS, or reactive oxygen species, inside red blood cells of mice. The young control rats, marked YC on the x-axis, represent healthy young animals without any plasma treatment. The old rats, marked OC, are untreated aged rats. As shown, they had much higher RS levels than the young controls, which is a typical sign of aging. However, after the treatment of plasma from young rats, the group labeled OYP, old rats treated with plasma, showed a decrease in RS, almost matching the levels seen in young controls. In panel B, researchers measured FRAP, which stands for ferric-reducing antioxidant power, reflecting the antioxidant strength levels seen in the young controls. Again, the OC group had weaker antioxidant defenses compared to the YC, but after receiving young plasma, the OYP group showed a decrease in FRAP levels, restoring antioxidant capacity to youthful levels. And shown in this figure, figure 2A, you can see glutathione, a vital intracellular antioxidant. It dropped with age in mice as shown in YC versus old C, young rats versus old rat controls. But it increased significantly after plasma treatment as the old rats treated with young plasma, their GSH levels increased significantly, almost restoring youthful antioxidant capacity. In panel B, we can see the plasma membrane redox system, or PMRS activity. Aging increased PMRS activity, but treatment of young plasma, OIP, further improved PMRS activity, suggesting better redox balance and stronger cellular defenses against oxidative damage. Altogether, these results suggest that young plasma can reduce oxidative stress and boost antioxidant defenses in aged rats. However, there are important caveats to consider. For instance, this was a rat study, and effects in humans may not be the same. Second, the researchers did not isolate the specific plasma components responsible for these changes, as in we don't know what in the young plasma caused these beneficial effects shown in the rats. Further research is needed before any conclusions about plasma therapy in humans can be made. As shown by these impressive research papers, we've seen that young plasma can have biological benefits in animal models. But before we get too excited, it's important to talk about the ethical side of science. In 2018, Hoffman published a critical analysis of blood-based anti-aging therapies, raising important concern about how technology can affect society. If young blood becomes a viable medical resource, young, healthy people could be pressured into selling their plasma, especially those in vulnerable financial situations. Hoffman also points out about access and fairness issues. Who would actually be able to afford young plasma therapies if they became real treatments? There's a really strong risk that these therapies would only be available to wealthy, potentially widening existing inequalities in healthcare. Beyond these concerns, there's also the question if we should treat aging as a disease. Just because science makes something possible doesn't always mean it's the right thing to do. The science is exciting, of course, but the ethics are complex. Because therapies like young plasma can move into human clinics, we need more research, public discussion, and stronger ethical frameworks to guide these decisions.